Welcome to This is Epigenetics, the first season of the bilingual podcast presented by Cirque's trainee comedy, where we interview renowned Canadian scientists about their career, their research, and more importantly, their passion for epigenetics. In this week's English episode, Sanne Janssen and Edward Gardner interview Carolyn Brown, professor in the Department of Medical Genetics at the University of British Columbia. We hope you will enjoy our last epigenetic discussion of the season.
Hello everyone and welcome to the podcast today. I am one of your hosts, Edward Gardner, a PhD student from U Ottawa.
Hello everyone, my name is Sanne Janssen and I'm a postdoc at the University of British Columbia and I'll be your fellow host today. So it is my great honor to introduce Dr. Carolyn Brown. Carolyn did her PhD and postdoc in the lab of Dr. Hunt Willard. She was moving across countries during that time from Toronto to Stamford and then to Cleveland, after which she came to the University of British Columbia, where she is currently a professor in the Department of Medical Genetics.
Thank you, Sanne and Edward.
Yeah, thank you, Carolyn. How are you today?
Very well, thank you.
That's great. So I guess the first question we have for you today is, what do you like to do to relax outside the lab, get out of the science world a little bit?
I am truly addicted to genetics, so I even find reading papers somewhat relaxing. But I guess maybe my favorite hobby would be to go cycling. And we've done some cycling trips. BC is a wonderful place if you like to bike ride. Not so much snow as the rest of the country.
So have you ever gone biking over to the mountains in BC or more of the flat roads there?
No, my husband and I like climbing the mountains too, so we'll do the North Shore mountains. We have hopes one day of going into the interior and going out to Revelstoke, but haven't done that yet. A couple of years ago, we went out to Banff and went up Highwood Pass, which is the highest paved road in Canada. So that was fun, a bit of a climb.
Oh, wow. That sounds really fun. So I guess coming back a little bit more to you as a researcher, what made you decide to go into research? I admit when I went off to university, I wasn't entirely sure what I wanted to do, but I always liked the idea of solving puzzles and looking at data to find answers that hadn't existed before. So I considered some different things, economics or environmental sciences, but really the biological questions of how do you make organisms? That just seemed to me to be the biggest mystery. I know each field says that they're addressing the basic question of all, but to me, the question of how you take a single cell and make an animal out of it, that's just fascinating. So that's what I ended up in. Yeah, I agree. That's fascinating.
I was quite fortunate. I went to fairly small undergrad university called Guelph, and we got to do research there as undergraduates. So I knew going into my PhD that I really loved the research. No matter how many times I failed, I had enough stubbornness that getting one answer out of a lot of fails was definitely worth it for me.
We have one question here. What do you think are the most fun aspects of being a scientist and getting into research? And secondly, what do you think are some of the less amusing parts in the same vein?
To me, the excitement of science has always been understanding something. And in particular, feeling that you understand something that wasn't understood before. So learning, I love, and learning to the point where you understand something, but actually discovering so that now something makes sense that didn't make sense before, and you can see a pathway as to how this happens. That's the thrill of science to me. On the flip side,
the thrills are not far between, but they certainly don't happen all that frequently, the big aha moments where you really feel that you understand something new. So it can be frustrating when you don't understand why an experiment isn't working. You put in all the reagents and you don't get a product. How come? But also that can be where the unexpected comes from. So having to keep an open mind while troubleshooting is a challenge. And then I guess, it's frustrating that there seems to be limitations with resources and you end up resubmitting grant applications and spending a lot of time in paperwork. And I think you just need to remember that all of that can improve the science by really sitting down and thinking through experiments and discussing them with colleagues. But sometimes it just gets so that you feel it's perhaps more frustration that you're on a bit of a treadmill trying to get things done. And the joy of science is the open-ended questions, but I guess also a frustration can be that you're never done. I'm still working on my PhD project. Every answer gives you 5 more questions, and that is both the joy and also the challenge.
Yeah, it is almost a blessing and a curse that it's really just a never-ending amount of questions we can ask.
And a never-ending amount of papers that you could read that might have clues. Yeah.
What would you say your favorite part about mentoring the students is?
I think there's 2 answers. And my absolute favorite would be seeing the excitement as a graduate student gets the breakthrough, has the experiment work, sees the result, puts the paper together. That thrill of finding something that wasn't known before and being able to share that with the student who's perhaps experiencing it for the first time. With undergraduate students, it's a bit more simple. It's just seeing them understand what they've read so that you can actually do an experiment, find an answer that, even though minor and doesn't actually answer big questions, is actually data that has not been seen before, that you can actually contribute to knowledge. So sharing those moments with the students.
Do you find that you see kind of bits of yourself in your students at times when you're thinking about your initial journey into the scientific world?
Yes, I certainly think I do, and I try not to really, because I think everybody has their own journey, and we shouldn't be biased by our experience in thinking that would be a good journey for anyone else. I do think it's a bit of a deficit in that many trainees are probably going to go on to careers other than staying in academia, and yet they're being mentored by people who are in academia. And so we do have this inherent bias that we think this was a great way to go. And that's what our experience is. We don't have the experience about going out into careers in industry or looking to start startups and things like that. And I certainly hear from many students that they'd like more opportunities to discuss those aspects, but the expertise of their mentors is often very limited because that's not the way they went.
Yeah, that's a very good point. So actually, what sparked your interest in epigenetics?
I think my interest in epigenetics was a natural one from how do you get a complex organism? Because humans, all of our cells are the same genome, a few exceptions. But why do you get a kidney and a liver and a retina out of the same cell? And that comes down to gene regulation, to epigenetics. You need to remember the instructions of what tissue you are. And so how do you do that? And to me, then X inactivation, where you tell one X to inactivate and then remember that decision through the lifespan of the individual, but also in cancers, you get this massive growth, millions, even billions of cells, and it's always the same X that has been remembered that this is the one that's marked to be inactive. So I just feel that X inactivation is a beautiful system in which to study memory and also choice.
So talking about X-chromosome inactivation, can you please explain to us what X-chromosome inactivation is?
So X-chromosome inactivation is the silencing of one X chromosome and that occurs in individuals with more than one X chromosome, so females generally. And that's necessary because males have a single X chromosome and the sex-determining Y chromosome. And while the X and Y have both evolved from actually an ancestral autosomal pair, once the Y chromosome acquired sex-determining function, there was selection to streamline that chromosome and prevent recombination with the X chromosome. And so most of the 1,000 or so genes on the X chromosome have been lost from the Y chromosome. That would mean a massive gene imbalance between males and females. And so it's addressed transcriptionally by silencing one of the X chromosomes in females. There's an underlying question there. If they were ancestrally two copies and now they're one copy, that too is a dosage imbalance. And so there's probably a dosage compensatory step which raises the level of expression from the X chromosome. But the process that we study is the one that's specific to females, individuals with more than one X chromosome, which is silencing of all but one. And so in aneuploids, when you get more than two X chromosomes, you inactivate all but one. There are actually up to 5 X chromosome cell lines, and 4 of those X's have inactivated.
Can you explain a little bit more about what Xist is and how it performs its functions within the cells for X chromosome inactivation?
So the inactive X chromosome expresses this gene, a long non-coding RNA called Xist, and it's named for its XI-specific transcription patterns. And Xist is transcribed from what will be the inactive X chromosome, but it never actually leaves that chromosome. So it's transcribed and coats the chromosome in cis. And we believe that, similar to other long non-coding RNAs, it acts by recruiting proteins. And the proteins that it recruits are the classic epigenetic silencers. So it recruits PRC1, PRC2, and eventually you get an inactive X chromosome that has all of the classic features of heterochromatin. And so when we talk about X inactivation and that initial choice, a lot of studies are focusing on how do you get Xist expressed only from one of the pair of X chromosomes in the same nucleus. And so that process is probably different between humans and mice, because in mice, it's been very well studied that there's an antisense that's transcribed across Xist, and
that's the regulator that's controlled, whereas in humans, that downstream region where the antisense starts transcribing from is not well conserved. And so it's probably 5' elements or more distal elements that regulate how Xist is expressed from the future inactive X chromosome. But we then see Xist expression through all somatic cells. So it's not just a developmental gene, it's ongoing, although once it's recruited all of these heterochromatic changes, for most genes, they are very stably inactivated. They've acquired DNA methylation and they don't reactivate if you lose Xist. But some genes do appear to reactivate, and the cells become more sensitive to other epigenetic perturbations, such as loss of DNA methylation.
One more question. When you were getting started, were there any professors or scientists you really looked up to that got you started on your research career and the questions you ended up asking further on?
Yeah, my scientific history is a little different. As Sanne noted in the introduction, I started my PhD at the University of Toronto, and then my professor, Hunt Willard, moved to Stanford, so I moved with him and finished my PhD working from Stanford. Then he moved again to Case Western, so I moved again. So he was really a huge influence on asking questions and the rigor of science. In terms of sort of scientific heroes in the field of X inactivation, Mary Lyon is a huge figure, and in fact, X inactivation can also be called lionization of an X chromosome. And to me, the fact that she could take observations and make this whole hypothesis was just spectacular. That she was studying mouse mutants, and the ones that were on the X chromosome, she observed that the females, like the calico cats in my background, the females had a mottled mosaic pattern. And so she took that and the fact that in mice, you can survive if you're a female, even with a single X chromosome. And the fact that it had been seen that you could have extra X chromosomes and the phenotype wasn't too severe, but that they formed this condensed bar body. She took those together and said, all X's but one in a cell are becoming inactivated, and that's what that bar body is. It's an inactive X chromosome. And I mean, she even proposed that not all genes on the X would be inactivated, that those that had Y homologs might retain their ability to be expressed from the X chromosome. So it seemed that when I started studying X inactivation 30 years after her hypothesis, I was just sort of finding molecular facts to validate what she'd suggested. So that was really inspiring.
Those are some impressive predictions. I mean, you didn't have all the technology back when she was probably first looking at that to really ask these questions with 100% confidence, I guess. Yeah,
it's amazing to think of a pre-genome time. She only had mutations, didn't have the sequence of the genes. There wasn't molecular analysis like there is now. I find that in teaching genetics, it's hard to have students grasp that we didn't always have a complete genome. You didn't find a gene by looking it up on a browser.
Yes, in that sense, it is amazing the progress that has been made over these past couple of decades. Just it's hard for me to imagine that we cannot go into the UCSD browser and type in a name and see where it's at.
And everything about it, what tissues it's expressed in, what its epigenetic marks are. It's just so much information now.
Yes. Now I'm curious, if you hadn't become a researcher, what do you think you would would be right now? Do you think you mentioned economics? Do you think you would have been an economist?
I think probably, and my lab will probably laugh about this, I think I would be a data analyst. I mean, I like digging through data. My lab would laugh because I haven't become a bioinformatician. I tend to email them questions. I'm not even really adept with Excel at finding the answers, but something like that, I think.
Would you mind explaining to the audience why a calico cat is interesting in regards to X-chromosome inactivation?
Yes, that's a great question. And my lab website, we feature a lot of calico cats, and I'm always very keen to point out we don't study cats. It's just that they really do make a great visual example because the coat color gene in cats, which gives you either orange or black, is X-linked. So that means that females have two alleles. They can either be homozygous black, homozygous orange, or in fact, the heterozygote is the most common. And that means they have black on one chromosome and the orange allele on the other chromosome. But X inactivation, in order to make the dosage of the X chromosomes equivalent to the males who only have a single X chromosome, inactivates one of those cells very early in development. And so that choice of X chromosome to inactivate is inherited, our epigenetics, stably across somatic cell divisions. So you end up with patches where orange has been inactivated and you get black fur, and patches where black has been inactivated and you get orange fur. So a calico cat is visually showing us the mosaicism that most females have for having inactivated their paternal X chromosome in some cells and their maternal X chromosome in other cells.
And does this actually also occur in humans, where in our body we have some cells that have the X chromosome from our father activated, and in other cells we have the X chromosome from our mother inactivated?
Yes, very much so. It's just it's not visually apparent, but if you look at carriers of X-linked disease, for example, if you do a muscle biopsy of a heterozygote or a carrier for Duchenne muscular dystrophy, you'll see that there are patches of cells that are expressing the protein and patches of cells that are not. For some X-linked diseases, then there's actually selection, so that the cells that have the mutation on the active X chromosome and aren't producing the protein, those don't grow as well, and you see skewing of X inactivation where the cells that end up in the adult are those that have the good copy on the active X chromosome. And we'll see that in carriers of X-linked immunodeficiencies. In their blood, you see complete skewing of X inactivation.
So in that case, it's our body trying to help us out.
Yes, yeah. So for the X chromosome, it's quite unique in that males get X-linked diseases with a single mutation because they're these hemizygotes. They only have one X chromosome, whereas the female has two X chromosomes, but each cell is expressing one or the other copy. And so if it's really advantageous to be expressing that protein, then those cells will be the predominant ones just by natural selection within the body.
Is natural selection just happening more in development, or is that continuing to happen a lot in the adult bodies as well?
So what is seen is that as females age, so throughout adulthood into the 80s, there will be more skewing of X chromosome inactivation in the blood. And so we think that probably is evidence of ongoing selection. There have been some big twin studies where they tend to see that two female twins will start skewing the same way. Further evidence that there's a selective advantage
to one or the other X being active. But also you can get skewing of X inactivation by chance because it's an early choice. And if you kept flipping the coin the same way, you would get a population where one X was more often active than the other. And sometimes there are probably early initial developmental events that reduce the number of precursors. So our colleague Wendy Robinson at the Children's Hospital has studied cases of confined placental mosaicism. There's probably an early trisomy that gets rescued, but in the embryo, you don't see the trisomy, presumably because those cells actually died. But often those individuals have more skewed X inactivation, probably because there were fewer cells that actually were diploid and contributed to the embryo.
Oh, that's really interesting there.
Yeah, so you can get essentially skewing all the way along from very early development right to older individuals. And then in mice, they've actually found that there are alleles that can influence which X gets inactivated. But we don't see too much evidence for that in humans. In big population studies, there doesn't seem to be much of a genetic influence on which X gets inactivated.
Is there any particular reasons you know that the case might be different for the mice, where you get those specific alleles that have a bit more of an influence on the skewing of the inactivation?
I mean, they were identified in different strains of mice. And so mice have a very long, they have a very short generation time. And so an evolutionarily long period of time in which they've been separated out into strains. And so it may just be that these alleles got captured in different strains, and then you could cross them together and see the skew. But it also may be just something about the genomic structure.
Talking about mice and human X-chromosome inactivation, what are some other differences between these two species when it comes to inactivating one of the two X chromosomes?
Yeah, it's a cup half full, half empty viewpoint. To me, studying human X inactivation, I think there are a lot of differences. So one topic that we study is genes that don't get inactivated. So these genes that escape inactivation and continue to be expressed from what otherwise is a silenced chromosome. And in humans, there are a lot more of those than there are in mice. So in mice, probably about 6% of genes fall into that category, whereas in humans, it's 15 to 20 to 25%, depending on where you cut the threshold for what do you call escape from inactivation, because a bunch of these genes are variable. So they don't escape in all people, they don't escape in all tissues. So that makes it a gradient as to how many genes there are, depending on what your thresholds are. So that's a big difference between humans and mice. We have more of these escapees. The master regulator, willing to let me call it that, of X inactivation is Xist, this long non-coding RNA. And as expected for a non-coding RNA without protein coding restraints, its sequence diverges across all species. And so it's probably only about 70% similar between humans and mice. So that's either 70% similar or 30% diverged, depending on which way you're looking at it. And then a big difference that is still being really resolved at the moment is in the early stages of X inactivation. So in mice, there's a very complicated process. The paternal X expresses Xist during spermatogenesis, and so it comes in probably pre-inactivated, but it
becomes active at zygotic gene activation. But then the paternal X is inactivated in all the extraembryonic tissues. And then there's a reactivation in the inner cell mass, and then you get random inactivation. In humans, the X in the extraembryonic tissues can be either maternal or paternal, the X that's inactivated. So we don't have this exclusive paternal inactivation that's seen in mouse extraembryonic tissues and actually seen in all tissues in marsupials. And we probably don't have this on, off, on, off. Instead, inactivation occurs later and is random in all tissues. But those very initial stages, it looks like maybe both X's get suppressed a bit and then one X gets completely inactivated. And so, of course, in humans, it's a very difficult time point to be studying right around the time of implantation, late blastocyst. So there's only a few studies, but we can also look in embryonic stem cells, but another difference. So in mice, embryonic stem cells are derived with 2 active X chromosomes. That's not a really stable state, but you can get them with 2 active X chromosomes. And then upon differentiation, 1 X inactivates. When you isolate human embryonic stem cells, they tend to be from a later state and already have an inactive X chromosome. So you have to push them back into a naive state. But if you just culture human embryonic stem cells from a female, that inactive X can erode. And so that's problematic for stem cell biology, and a lot of intermediate steps there that need to be worked out.
Okay. So there's also some genes that are able to escape from the inactivation process. And I believe there's some differences between mice and humans. Can you elaborate on that a little bit more?
Yes, so in mice, there are fewer genes escaping inactivation. And in humans, some of those genes that escape in mice also escape in humans, and they also actually have gametologs on the Y chromosome. So in males, you're getting expression from the X and Y, and in females, expression from the X and the inactive X, or the active X and the inactive X. But in humans, if you look at those genes, the genes next door also appear to be escaping inactivation. And so whether those are bystanders who haven't been suppressed yet, or whether those two share signals to escape inactivation, that's the sort of research that we're currently doing to see why genes escape inactivation and why is it the same rule for all of the genes that escape inactivation?
That's a very interesting question, yes.
So it's been argued that the genes that remain being expressed as housekeeping genes from the Y chromosome, so the Y's acquired a bunch of testes-specific genes, but it still has a handful of genes that were once on the X chromosome and are still retained on the Y. And that may be because they are very, very dosage sensitive, and you couldn't go through a transition of losing a Y copy and suppressing an X copy. But evolutionary arguments are always hypotheses.
Yeah, they're very difficult to test generation times.
And particularly if you want to know what happened in humans.
Yeah. So these extra adjacent genes which escape, do those often have homologs on the Y chromosome too, or is those sort of giving the female cells an extra bit of those gene copies?
Yes, so those have lost their Y homologs, and other genes along the X chromosome that escape as well have lost Y homologs. And so we do think that there's actually more expression in a female than there is in a male. Even for those with Y homologs, it's probably not perfect dosage compensation, but we don't know always whether the Y homolog or gametolog is equivalent to the X. They have acquired changes over time. But in general, the Y expresses those genes more than the inactive X does. So in those cases, males are expressing more, whereas the X escapees without Y gametologs are expressed more in females. But in addition to being expressed more in females, which of course could contribute to sex differences, the ability to express from both X chromosomes may also offer a bit of protection against mutations in cancers. So there have been some studies suggesting that those genes that escape inactivation are drivers of cancer in males, but not so much in females, and may lead to the male increased prevalence of particular types of cancer.
So being involved in the discovery of Xist, especially back in those days, how did you find Xist?
So very early, for my PhD, I was studying the process of X inactivation, and I was actually had two projects. One was to identify what we call the X inactivation center. So this is a region of the X chromosome that is required in cis in order for the chromosome to be inactivated. And we knew there was such an element because of X chromosome rearrangements, that only when a particular region was retained by that X chromosome could the chromosome inactivate. So these were karyotypic studies that clinicians had done on patients with X chromosome rearrangements. And so in the lab, we'd aggregated many of these, and we're trying to refine the map of where this single region could be. And there were so many rearrangements reported in the literature that we could map it to a 1 megabase region. Again, before there was an assembled genome, just by southern blots with probes. And the other project that I had was to look at genes that failed to be silenced on the inactive X chromosome. And so for that, I was using somatic cell hybrids, mouse cells that had been fused with human cells. And then when you do that, human chromosomes tend to be lost. We had a mouse cell line with a mutation in a gene that escapes inactivation. And so by growing these cells at high temperature, you would select for either the active or the inactive X chromosome to be retained in this hybrid. And then we'd select against the active X chromosome because it expresses genes that you can use chemical selection for. And so we had hybrids that had an active X chromosome or hybrids that had an inactive X chromosome. And then we'd actually do northern blots looking for whether genes were expressed from both or only from the active X chromosome. That was the start of studying these genes that escape inactivation, and we found that there were far more than we'd been expecting. Those two studies came together then when we got a gene which we thought mapped somewhere else, which actually mapped right into that X inactivation center interval. And that gene, rather than being expressed from the active X, it was only expressed from the inactive X chromosome. So that was the first gene we'd ever seen that wasn't expressed from the active X chromosome. And in hindsight, that rather make sense to have an RNA that never has to leave the nuclear environment as a controlling element, because it can be right there on the inactive X chromosome. But yeah, it took a lot of convincing. And in addition to the unexpected nature of having an RNA, it was only the second real long non-coding RNA that had been described. So H19 had just been described by the Thielman lab. So it took a lot of convincing of ourselves that there wasn't an open reading frame in that gene. And it's a long gene. So it's 17 to 19 kilobases alternatively spliced. And this was just at the advent of automated sequencing. So that was a lot of sequencing that had to be accurate enough to know that you didn't have an open reading frame.
Yeah.
It sounds like a lot of an initial challenge there.
Yes, but very exciting.
Did back then, did you, when you found Xist, did you realize how important this RNA would turn out to be?
I think we were pretty suspicious right from the beginning that why would something be expressed only from the inactive chromosome other than to inactivate? And it was in the critical region that is required for inactivation. So those two facts together were certainly pointing in the direction that it was critical. The lab collaborated with Jeannie Lawrence's lab, who are experts at in situ hybridization, and they did RNA in situ hybridization and showed that the Xist RNA was just coding that inactive X chromosome, and you saw no RNA from Xist over on the active X chromosome. So there were a lot of smoking pistols.
That must have been so cool to actually see that for the first time.
But there were also lots of scares, if you think, oh, but it's heterochromatin. Maybe it's just that the RNA sticks. That's why we're seeing it from the inactive X chromosome. More controls.
I think it turned out to be more than worth it to do all those controls.
Yes.
So once you had all those controls down, you're absolutely 100% confident that this was the real thing. How did you feel at that moment?
I think it's like so many scientific moments. There isn't a single moment. I certainly remember the first northern blot being kind of suspicious, was just at the advent of doing RT-PCR, not qPCR, RT-PCR. And so then doing that with panels and thinking, yeah, this really looks like it's only from the inactive X. And then months of these controls and sharing the results, presenting the results. I don't think there's one moment where I think it's just growing faith. But there's all sorts of odd things about the history. So how did we actually, where did the gene come from? I told you that we looked at the gene's location, but it was what used to be called clone by mail. So a colleague sent us a clone that they had isolated from an expression library. So in theory, this was supposed to be a gene that was expressing a protein, but we never found the open reading frame, so we didn't think it made a protein. But it always niggles at the back of your mind. Maybe there's a small protein somewhere in there. And, across the decades, more and more data piles up to support you. But evolutionarily, it looks like Xist actually derived from a protein coding gene. So at one point, there was a protein coding gene that lost its function and acquired additional domains, allowing it to become an activating long non-coding RNA.
For the younger generation among us, would you like to explain what a northern blot is?
Northerns and southern blots, yes. Without a genome, you don't have sequence of what you're looking for. You can't make PCR primers. So you would radioactively label pieces of DNA that were derived from the genome by some process. So there were libraries that people had made by fragmenting the human genome or by trying to, and you could screen them with other DNA to try and get flanking DNA, or you could screen them with antibodies to try and get expressed regions of the genome. And so we were interested in the X chromosome, and the Willard lab would get sent probes that people pulled out and identified were on the X chromosome. And so we would then try and map those to the X chromosome, and then any that were in this region of the X inactivation center were the ones that we were particularly interested in. And so for expression, it was even harder because your RNA doesn't last all that well, but you'd run the RNA on a gel and then hybridize it with one of these probes, and it should light up the RNA that corresponded to your gene. And you could then look across tissues and see where is it expressed. But Xist, being 17 to 19 kilobases in size, was very, very large. And so you had to have very good RNA in order for it still to be intact enough to see a band that big. And again, it wasn't that convincing a band. Had it been down, a nice 2 kb band, it probably would have been much more convincing, but this was right at the top of the gel for resolution. So less convincing that it wasn't just some background smear that had got stuck up there. So yes, it was nice to see the advent of PCR and stop having to do so many northerns and southerns.
Yeah, those always seem like a very time-consuming process.
Yes, I don't think today's young scientists could handle the idea that you did your experiment and you clamped it all closed and you put it in a -70 freezer for, let's say, 2 weeks to get the X-ray film exposed, and then you develop it and think, man, I should have left it longer. But you had to consider that your radiation was also decaying with a half-life of 2 weeks, so leaving it longer wasn't gaining you as much as you wanted. Yeah, they were very long experiments.
Wow, yeah.
Whereas now you'd do a PCR and have your answer the same day, with a lot less RNA that didn't have to be as intact.
Yeah.
So the discovery of Xist can really be called a serendipitous experiment. We were anticipating that this would be another gene that was protein coding that would map maybe to our region of interest and should be expressed from an active X and maybe an inactive X. Those were the questions we were answering. Instead, we found something that was expressed only from the inactive X chromosome. And so, well, it was very lucky. I think it's a good example of luck favors the open mind, because you need to look at those experimental results and see what they say. And these said that this was an RNA expressed only from the inactive X chromosome, which had not been seen, but obviously was and was going to be very interesting.
Yeah, that would be. So for studying the X chromosome inactivation in your lab, what kind of techniques do you use in your work and the work in the lab?
Yeah, so for studying the Xist RNA, we still use a lot of RNA in situ hybridization because that lets you see where Xist is.
And for studying the epigenetics, then you can see which marks co-localize by immunofluorescence. So that's certainly a big line of experimental approach. In terms of our model system for studying Xist, much of our work has been done using a system that Jen Chow, a former grad student in the lab, developed, where she integrated an inducible Xist cDNA into a male cell line, and then using an inducer, doxycycline, you can turn on Xist and then see what happens when you turn on Xist. But this is a model for human X inactivation, but it is using a somatic cell rather than the developmentally appropriate embryonic cell line. So we use that a lot for studying escape from X inactivation. A student in the lab, Sam Peters, in collaboration with the Wasserman and Simpson lab, integrated a human back, so an artificial chromosome of 150 to 200 kilobases of DNA, from a region that escapes inactivation in humans, but not in mice, and integrated that onto the X chromosome of mice, and saw that that same region escaped inactivation now in mice. So these human signals that say this gene shouldn't be inactivated are conserved. And so that's an approach that we're using. She's adapted that system to embryonic stem cells to try and identify what are the signals for the genes that escape inactivation. And then, because there's so much epigenetic data available, another student in the lab, Brad Balaton, he has data mined all available data, because often the X chromosome is thrown out of studies. If you look at genome-wide studies, often they go from chromosome 1 to 22, and the last 5% of the genome's completely ignored, because if you analyze it, you have to analyze it in a sex-specific fashion, which probably wouldn't be a bad idea anyways. But also the dose is different between males and females, so it is difficult data to analyze. But we've reanalyzed a lot of data from epigenome-wide studies to see what's going on the X chromosome, in particular, what differs between genes that are silenced, genes that escape inactivation, and then how come some genes are sometimes subject, sometimes escape. Those in particular are very interesting because it's the same genomic environment, but something causes that switch to be different.
It's very interesting. The gene switch sometimes escape and sometimes don't. A lot of that is tissue specific or environmental specific. Do they have specific functions for some tissues or specific ways of evading some of the stressors that people would maybe have in a uterine environment or other situations?
Yeah, so the tissue specific escape is definitely an area that's being explored by a lot of groups right now. For us, what we're interested in are not genes that escape inactivation and are only expressed in one tissue. We don't consider those tissue-specific escape. We're interested in the genes that are expressed in multiple tissues, but are silenced in many and escape in another. So many of the genes with specific functions for a specific tissue are in fact expressed only in that tissue. And so we don't know if they're meeting our definition of tissue-specific escape. But some of them probably do have a particular role when they escape inactivation. A number of labs are studying some of the genes related to immunity, and they seem to reactivate under disease conditions in some of the B and T cell lineages. And that may be related to how Xist is retained on that X chromosome. But that variability may underlie some of the female predisposition to autoimmune diseases.
Where do you think your field of research is heading in the coming years?
Yeah, so I think we still have a lot to learn about how human X inactivation is controlled, how that differs somewhat from the really optimal mouse models. And I think other groups, we haven't been participating in this, but are developing better models to actually study human X inactivation at the time of inactivation and differentiation. We've been using our somatic cell models and are continuing to use that to try and identify which regions are critical for X inactivation. We'd like to know whether there are regions that are absolutely critical and could be impacted by mutations, and other regions that are less critical. And then for escape, we'd really like to know what are the elements that control silencing. Are there genes that are more susceptible to reactivation? Are there pathways that make genes more susceptible to reactivation? How does the DNA genomic context, the RNA and associated factors that are recruited by Xist come together to actually control expression from the inactive X chromosome? And I think Xist still has lots, Xist and X inactivation still have lots to contribute to understanding epigenetic gene regulation. What we're seeing is very much that it's this interplay of factors that you need to understand more than the DNA, the long non-coding RNA, the proteins. You need to understand them all together in context, and then in context in the nuclear ultrastructure.
If you could give your first year PhD self an advice, what would this be?
I think I would tell myself to be more proactive about career mentoring. I would probably tell myself to be more focused in my experiments, but I'm not sure that that would be good advice. Some of my side projects have become favorites. So, and I think I'd tell myself not to be daunted by the setbacks, that it's all part of the journey. But I think the fact that I made it to the end where I'm looking back on that question suggests that I did okay with that.
So that was it for today's episode. Thank you all for listening to the podcast. Of course, a big thank you to Carolyn for being here with us and sharing your expertise and providing all this valuable advice.
Thank you both very much. And I think it'll be a great series.
Thank you. It's excellent talk you gave today here.
Thank you for listening to the first season of This is Epigenetics. For more informative content about epigenetics, visit our website and stay tuned for more news about Season 2.